You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast. I have Dr. Ronald McGlennen, founder of Access Genetics and Oral DNA Labs. We're going to be talking about the oral microbiome and periodontal disease and other conditions of the mouth. So, Ronald, thanks for coming.
My pleasure. Thank you for having me this afternoon.
Yeah, I mean, even the microbiome is niching. Some are gut people, some are oral people. Why the interest in the microbiome, and why specifically the oral part of it?
Well, I think firstly, in medicine broadly, we've come to recognize that the microbiome, whether it's in the mouth, in the gut, or on our skin, now duly has to be recognized as an organ in our body. It's essential to health. And we have seen in the last few years that the embrace of understanding the microbiome and those organs or those areas of the body very much dictate and have provided solutions to diseases that previously were really difficult to manage. We'll talk about the oral microbiome in a moment, but if you just think about changes in the gut microbiome, explanations for inflammatory bowel disease, Crohn's disease, and other conditions have been really difficult to treat. And now we have solutions where we focus not on the intestine, but we focus on changes in the gut flora. The mouth, on the other hand, is, of course, an entry point to the body. It's actually on the outside of the body when you think about it. So there's continuity between the outside world and the human body at the interface of the mouth. So to a great extent, the bad bacteria that we would be concerned about come from our environment. We talk about the exchange between humans through intimate contact. We talk about the exchange of bacteria that come off foods and, of course, just off of surfaces in our environment. So the presumption is that when we're young, when we're healthy, we are in a state of eubiosis. That word means good state of life. And over time, through bad habits in our behaviors, our diet, our lack of exercise, and just the fact that we're getting older, we progressively shift to a state of dysbiosis or bad life. The microbiome is a reflection of that. The microbiome is a compilation of many, many species of microorganisms, and for the most part, in the state of health, the good bacteria predominate. The bad bacteria are either suppressed or non-existent. Over time and under conditions of stress or coincident disease, and we'll talk about a couple examples in a moment, we start to see the emergence of the bad bacteria. We'll use the word pathogen to describe those. So we can strive to look at the microbiome in its entirety, or we can frankly look at just the bad bugs. And if we can measure those bad bugs, and I use the word bugs as a synonym with bacteria, then we start to see where diseases start to occur. Now, there's actually a lot of disease. Please, go ahead.
Quick question. Okay, so in the gut, for instance, like C. difficile, supposedly it's there all the time. It's just in low numbers. And when there's a dysbiosis, then it rises to power. But was it a pathogen, or was it just there the whole time, but in small numbers, maybe it produced metabolites that weren't harmful, and maybe it was commensal? I mean, with the oral microbiome, is that what's going on? Like, there's no such thing really as a pathogen. It just depends on who gains power and and perhaps that microbe doesn't produce, metabolites and interact in a positive way with the community in the mouth?
Well, there's a simple answer and a more sophisticated answer. The simple answer is we believe that for the most part, these bacteria are resident in our mouth, in our gut all the time. They're just at low levels. And so whatever the bad effects of those bacteria are, they're suppressed and overwhelmed by the coincident presence of neighbors who are good bacteria. It's probably true that in the case of oral pathogens, they came from the outside. So we're not born into this world with a mouth that already has resident bacteria. They come from our parents, they come from our environment, and so we're seeded, and the microbiome becomes inoculated, if you want to use that term, with those bacteria. So they are there, and disease emerges typically when there is a suppression of the good bacteria, then thereby facilitating or allowing the bad bacteria to grow out. And so we can see this in serial test measures that we perform. A younger person has lower levels of these bad bacteria, and 5 years later, they're at slightly higher levels on average, and so on and so forth. And we've actually developed some stochastic models that show that patients who look normal in the dental examination start to show the early signs of periodontal disease, but well before you can see it with your eyes, we see that very purposeful shift from good bacteria to bad bacteria. And at a certain point, they get to a threshold level, and disease is present.
That's great. Yeah, that's good to know because it's early detection, and perhaps there's an intervention that can be made to bring them back to a healthier state.
Well, that becomes a kind of a policy issue, and to a great extent, dentistry, not different than physician-based medicine, is not today a focus on prevention. It's more show up with a disease, and then we treat the disease, and then we try to manage it. But we have some programs in place, and they're in the form of basic research that is searching to or striving to figure out ways to make predictions who is going to get disease and who will not. And we're using simple tests just to measure bacteria. A lot of bacteria means that person has disease. Less bacteria means that they don't. And what are those threshold points that we can then point out, publish, and say, look, our goal is to treat young people, keep them out of harm's way with disease by keeping these bacterial levels low.
In terms of interventions, have you tried any? What was the intervention and what was the effect?
Indeed. So one of the things that is true about dentistry is that the toolbox by which dentists reach into and treat periodontal disease is rather limited. The gold standard is something called scaling and root planing, and this is a process of mechanical removal of the biofilm. The biofilm is the term that describes where the microbiome resides. It's a physical entity that's stuck to the surface of the teeth or the epithelium surrounding the teeth. So that's the main tool in the toolbox. But then in the past, people have used systemic antibiotics. They have used local antibiotics. More recently, they've used a variety of chemical hygiene products. So you can think of mouthwashes. People have turned to lasers. And I would add, most recently, people have turned very intelligently to the idea of changing the microbiome by changing the composition of the bacteria, essentially replacing the bad bacteria by overpopulating with the introduction of good bacteria. Think of yogurt, for example, how yogurt is there to soothe the stomach, soothe the intestinal tract by replacing it with lactobacillus. Well, we can do similar things through the use of probiotics. Further beyond probiotics is the fact that there's something called a prebiotic. Bacteria, just like ourselves, have preferred foods. So if we change the nutritional component that goes onto the microbiome, we can actually shift from the bad bacterial growth to the good bacterial growth, thereby essentially treating the patient by just changing the biofilm, changing the microbiome. So we don't personally, in a clinical laboratory, engage in that type of therapeutic treatments. However, we do the testing for many clinical trials in a number of small studies. So we are privy increasingly to all kinds of innovations in the way in which we treat oral diseases such as periodontitis.
Yeah, that's great. Well, when you say you're characterizing the oral microbiome, or I'm using that word, what are you looking at? Are you looking at just the prevalence of species, with whole genome sequencing, or just 16S? Are you looking at metabolomics? Are you looking at transcriptomics? Like, how deep are you going, and what are you looking at? And maybe we'll get into why those are important or not.
Sure. So the answer to the question is we're doing all of the above, but for routine and daily and clinical applications, we are using a very precise, very accurate quantitative detection of the bad bacteria. We look at a profile today of 11 of those worst actors. Now, what's the justification for that? Well, firstly, we know that the presence of these bacteria at various levels or various quantities can be tightly correlated with disease severity. So the dentist has essentially a reference point that says, if I see some abnormalities through a visual examination, I can marry those up with these bacterial levels, and they can then classify that patient in terms of mild, moderate, or severe disease, and they can make their treatment choices accordingly. In terms of more advanced testing that's under development but not currently available for routine or daily clinical application, the analysis of the oral microbiome looks at a scattering of the good and the bad bacteria and looks at the relative levels of those two. We're still working to establish what are the normal or the reference ranges for those respective levels. So that's a tougher project, and I think others in this space, others doing research in the oral microbiome would have to agree that we are somewhat hampered in not really knowing what is health versus disease. So the analysis of the entirety of the oral microbiome is still a bit in its adolescence. So today, there's a heavy reliance, whether through clinical trials or through the rank and file dentists using our tests, just to measure the bad bacteria. And if I can borrow the analogy of the canary in the coal mine, we use canaries because they're very sensitive indicators of, noxious gases in a coal mine, for example. Well, we can use the serial measurement of these pathogens as a very sensitive and predictive marker of where that patient is on disease versus health.
Are the pathogens even the same species or strain in different people? I thought that a healthy or a non-healthy microbiome can have different actors in different prevalences and still be either healthy or not healthy. Where have you found that these 11 strains, yes, they're either there or not, and that indicates disease or not?
Well, your question is actually a fairly sophisticated one. So I think the first part of the question would relate to whether or not looking at 11 pathogens are representative of all of the pathogens. So are there 12th, 13th, 20th, 30th pathogens present, and are they different between individuals? I can answer that in part by saying that after doing nearly 250,000 tests on patients, the profiles fall into very predictable patterns. There's quite a variance in those patterns, but almost every patient showing disease has some representation of these 11 pathogens. So they are good indicators of disease. Now, the second part of your question is sophisticated in so much as some of the selected bacteria described as being pathogenic actually represent at the subspecies level a combination of good and bad actors. For example, there is a bacteria referred to as Fusobacterium nucleatum, or FN for short. Essentially, everybody has this in their mouth. And when disease comes upon a person, for example, when a person develops diabetes, FN will rise to higher levels. So in the state of health, we refer to this bacteria as a commensal. It lives in a state of peace or non-disease with that individual, that host. But when stress is upon that individual, FN will grow to high levels. It becomes pathogenic, and it can affect organs throughout the body. Well, looking at FN, Fusobacterium nucleatum, is insufficient, we know. No. That in fact there are subspecies, typically between 5 and 8, that reside in a given human. And knowing the ratios and the proportions of those subspecies is an emerging area of testing interest. It is not to say that any single subspecies is going to offset the effect of the bad actors, but we do believe that people have different clinical courses and hence disease severity based on the relative proportion of these subspecies. So we're moving not only in the direction of looking at more bacteria species, but actually having to go down to the subspecies level. You asked the question about looking at DNA sequence, and indeed, quantitative polymerase chain reaction, as an example, is essentially looking at the sequence level. The primers are designed to be species-specific and sequence-specific. Next-generation sequencing is on the horizon for routine clinical use. Its clear detriment, however, is that it fails to be sufficiently quantitative to be useful clinically.
Well, what even defines something as being pathogenic or not? Is it the metabolites it produces that have a negative effect on, our somatic cells? Is it that, certain pathogens actively outcompete beneficial bacteria? Like, what's the definition of pathogenesis?
Well, the simplest thing to think about is a wound on your finger, but I won't use that example. But the interface between our immunity, our immune response, played out through inflammation, swelling, redness, bleeding, tissue destruction, those are all manifestations of the pathogenicity of bacteria. Bacteria, like our own cells, produce proteins. Those proteins are secreted across the cell membrane, and those proteins in the surrounding environment will either affect the immune cells, i.e. causing them to be suppressed, or those proteins may actually act on the local tissue to cause tissue destruction. We see that in the form of a wound, what we call pus, right? That's dead tissue that's being dissolved by a proteolysis of both the combination of human cells and the bacterial cells. So these proteins are collectively referred to as virulence factors. And we characterize pathogens based on our understanding in basic science and basic culture experiments of what these virulence factors are. P. gingivalis, Porphyromonas gingivalis, is a particular bacteria that's being much discussed in science and, frankly, in popular science. Why is this important? Well, P. gingivalis is probably one of the more august pathogens that cause periodontal disease. Local inflammation in or around the tooth, the gums, causing bleeding, tissue destruction there. But those P. gingivalis can also leach into the systemic circulation. And when it does so, it can find residence in the heart, the pancreas, the liver, and more recently it's been described how P. gingivalis actually makes its way to the brain. P. gingivalis produces, as one example, a virulence factor called gingipains. This is a protease. It's a protein produced by the bacterium which dissolves local proteins. And gingipains, when they act upon human tissues, can cause the resulting degraded protein to be misfolded, and it has been hypothesized that gingipains are a cause of neurofibrillary plaques, which are characteristic of Alzheimer's disease.
Yeah, I've heard that certain bacteria in the mouth are showing up in atherosclerotic plaques, in the blood vessels and, like you said, in various other parts of the body.
Mm-hmm. That's correct. So to cite the example that I use, we collectively refer to the effect of oral infections on systemic disease. You cited the example of atherosclerosis. Live bacteria have been identified within the atheromatous plaque within the arteries of the heart. Some of these bacteria are actually viable, so they can be retrieved and grow in culture. But it's believed that these bacteria, when they localize to distant sites, cause or evoke inflammation. So when we talk about heart disease, it's not just about the deposition of lipids in the coronary arteries. It's the inflammation associated with that lipid deposition. And it's believed that the inflammation is caused by the effects of these bacteria. Follow that train of logic at any organ in the body. The localization of oral bacteria in the beta cells of the pancreas cause those beta islet cells to become inflamed. That leads to a relative state of insulin deficiency, and that is believed to be a source or a cause of type 2 diabetes. And you can basically go throughout any organ in the human body and think of a chronic disorder, and you can start to construct a model where oral bacteria are involved in inciting chronic inflammation that then leads to one of the large killers of humankind: heart disease, diabetes, Alzheimer's, and yes, even cancers.
So it sounds like preserving the integrity of the mouth and obviously the whole digestive pathway of the gut, all the way through is critical. But, sticking with the mouth, it sounds like, again, preserving the integrity of the mouth is critical. Otherwise, it allows all kinds of these bacteria to migrate to spots where they, take up a pathogenic role where they shouldn't be, essentially.
Yes, and if anything, it turns our attention away from interventions where we're actually trying to keep those diseases at bay, and as we talked about earlier, to focus on finding the earliest indications of that disease and stopping the process before you start to see overt clinical signs. So campaigns that start with people in their adolescence and start to look to see, do you have the early signs of periodontal disease? When you do the math, with almost 100 million Americans involved with periodontal disease, and you start to translate then its effect on heart disease, diabetes, Alzheimer's, you see that there is a real need to start acting sooner. And so the cause and the charge is on to dentistry to start really embracing prevention as opposed to just treating established disease.
And, maybe it's obvious, but what is periodontal disease? What will the average person experience and say, they'll be told they have periodontal disease, but what are the, what happens in the person? Do their gums bleed? Like, what are the signs of it?
So periodontal disease, the periodontal tissues, by description of the word, are the tissues that surround the base of the tooth. The tooth is embedded into the jaw bones. There is a ligamentous tissue that supports that tooth, and then the soft tissue, the gums, surround the tooth. When there is pockets of infection down below the gum line that involve these varieties of bad actor bacteria, then we have what's called a polymicrobial infection down below the gum line. Those diseases or those infections are very subtle. Most people do not complain of pain, so that's not what drives them into the dentist. In fact, pointedly, they're much more likely to go because their teeth are just colored than because they know to have gum disease. Now, they might be forewarned if somebody says, gee, you have bad breath, or halitosis is the medical term. So that might be a reason to go in. But usually periodontal disease is asymptomatic. Now, when they floss their teeth, if they do so, they will see that the bleeding occurs. The bleeding that occurs is a manifestation of the inflammation, which is the body's response to this infection. Bleeding is due to the blood vessels are being very friable and delicate, and so when the floss goes down there, it fractures those blood vessels, the bleeding results. That's a byproduct of the fact that there's a lot of swelling, a lot of inflammation, some local tissue destruction. That's a hallmark that a patient can see at their home while brushing their teeth, while caring for their teeth at home. The dentist sees this and takes an x-ray. If the x-ray surrounding the tooth shows loss of bone and bone support around that tooth, that along with bleeding, that along with the physical or the visual observation of swelling are the clinical signs. We enter in the scene with a test that measures the type and quantities of the bacteria which are evoking that inflammation and causing that tissue destruction. So we would like to argue that x-rays and testing and the visual examination are three essential components of making the diagnosis. That's what periodontitis is.
And has it been observed in people that, they brush, they use like in between toothbrushes, I don't know what they call them, they floss, they take a lot better care of their mouth. At first it seems like that would cause a lot of bleeding, but I would guess perhaps those vessels that are near the surface that you said are friable heal up, or
that's true.
I guess what I'm asking is, what are ways for people to make an intervention to help themselves?
Well, you make a really good point. It's probably true that it's insufficient simply to brush one's teeth. Flossing, although there's been some debates in the popular literature that it may not actually add much, that plus other types of mouthwashes will help to suppress or keep those bacterial levels at lower amounts. But the simple fact is that the flossing helps to break loose or mobilize the buildup of the plaque-like material that's below the gum line. And by mechanically removing that, you've given a head start on the effectiveness of your toothbrush and of your mouthwashes. So the floss is important to get between the teeth and liberate that hard plaque material, which really is nothing more than sort of a concretized version of this mass of bacteria. So we're debulking the amount of bacteria in the mouth when we floss.
So these bacteria that cause periodontal disease, I guess they're trying to set up their own microenvironment, their own niche in which they can flourish.
Mm-hmm.
Is that what happens? Is that what plaque is, for instance? It's just a hardened shelter for these kind of bacteria where they can hang out and multiply?
So we started off with the terms the microbiome, and for simplicity we'll say that this is the aggregate of the community of many species of bacteria. But how do they actually live? And so in the case of periodontal disease, they live below the gum line. That environment is largely devoid of oxygen. So it's an anaerobic environment. So that pre-selects those types of bacteria that can tolerate an anaerobic condition. This community is actually very sophisticated. There are layers or strata. There are certain bacteria that love to live adjacent to each other because the metabolome of one species might actually be the nutritional basis of another species. So when we speak of a eubiosis, we're talking to a great extent about the predominance of streptococcal species that are anaerobic. Those are the good bacteria. And then they're seeded, as I suggested earlier, with a smattering of these bad bacteria. If you change the health conditions, i.e., if you have a lot of sugary foods, if you have a lot of other types of dietary components that sort of foster the growth of the bad bacteria, you'll see the shift from the predominance of the good bacteria to that of the pathogenic bacteria. This all occurs in a tightly interconnected community. And when you look at this under a phase contrast microscope, for example, you can actually see structure. There are bacteria that live on the surface of the plaque, and there are others that live closer to the surface of the tooth or to the epithelium. There's those that create a walled-off perimeter that serve as a nice barrier. The simple fact is, if you know what the concept of an abscess is, then these plaque that live on the subgingival or below the gum line areas of the mouth are very analogous to that. And when that becomes hardened with some calcium, that's what plaque is when we physically remove it.
So in one way, could you consider everything you eat to be a prebiotic, just for either the good team or the bad team?
I'm ill prepared to answer, what are the favored foods of pathogens versus the good bacteria. But suffice it to say, they're looking for similar things. But let's just use one example. I referenced Porphyromonas gingivalis. Well, they love iron-containing foods. And one of the byproducts of the breakdown of red blood cells is something called hemin, H-E-M-I-N, and that is of nutritional support for P. gingivalis. So inflammation leading to bleeding, exacerbates the growth of P. gingivalis, as an example. But as I say, almost anything that is part of our nutritional requirements probably has some effect on the microbiome in the mouth and, of course, in the gut.
So what you said you were able to sequence what's going on with the oral microbiome in many different ways. You know, what metabolites they create, I guess what genes that the species have, what genes that they express. I mean, what proteins that they give off. I don't know what's proprietary and what's not, but what, I don't know, interesting correlations or things or methods have you found so far where people can make interventions to improve someone's health if they have a problem?
Mm-hmm. Well, what we have sort of put together is a profile for a typical patient and, quite frankly, for a typical type of therapy, where we ask the question, if we take the 11 bacteria that we measure in our quantitative DNA analysis, and we sum that to represent a cumulative bacterial load. So think of it as in the form of a mass or just an overall quantity. And we ask, what is the effect of a first-line therapy, such as scaling and root planing? Well, we can start to quantify the typical, the extraordinary, and then the insufficient response, i.e. in terms of bacterial load reduction. We also can then start to characterize when is the best time to measure the absolute decline or absolute nadir of that amount of bacteria. We can then compare that to the effect of scaling and root planing when it's adjunctively used with antibiotics or with a laser or with prebiotics. So we can see the effects of primary and adjunctive therapies on reducing that bacterial load. Now, left to our own devices, after we leave the dental office, we know that those bacterial levels will start to slowly grow back up to those pre-treatment levels. And picture, if you will, sort of an open smile line where if over the course of roughly 13 to 15 months, a patient presenting with periodontal disease, treated by that dentist, then measured again at approximately 8 to 12 weeks, and then again measured at 3, 6, 9, and 12 months, we can pretty much determine the natural history of that bacterial, that complex bacterial set of infections. That profile will differ slightly between patients depending upon their baseline set of health. For example, the regrowth pattern will happen faster in patients who are diabetic or who are under chronic stress or who are immunosuppressed. The solution to that would be more frequent recalls to see that patient more regularly and earlier because you're going to want, again, to suppress those bacterial levels. We can see who's doing a good job in applying those therapies because we know what the statistical average is for the bacterial load reduction. So we can put this in the form of a patient-specific ideogram that hopefully in the future will be useful to clinicians to guide the frequency and the intensity of therapeutic choices, all driven by just knowing what bacteria are present and how successfully we can reduce them.
Okay. And then your clients are dentists or periodontists or individuals? I mean, who do you work with and what kind of products do you have for people and for labs?
We are a clinical laboratory, so our focus is the management of a collected sample in a dental office or a physician office. And then we produce a test result, and then we provide a lot of consultation as to what those test results mean. And through my voice or those of my teammates on the customer service side, we provide a lot of value to helping people know when to add extra treatment modalities, for example. But you ask an interesting question because our client base, the customers that we serve, are mostly dentists, some of which are periodontal specialists, but increasingly, and I'd say day by day, the number of calls we receive for new clients are physicians. Physicians are eager to understand why their patients have heart disease, Alzheimer's disease, diabetes, and they have come to know that it's not just about measuring in one case cholesterol or glucose in another. It's about understanding if that patient also has good oral health. The biggest challenge is connecting the physicians and the dentist together. And we do the best we can to facilitate exchange, collaboration, consultation. But you'd be surprised the number of physicians who are ordering our test, and with that result, they look in earnest to see if there are local dentists who can take care of their patient to help improve their heart disease, improve their diabetes, improve even their cognitive measures.
Yeah, I'm looking at the website. It says you have featured tests like MyPeriopath, early warning detection for periodontal disease, alert 2, oral risk HPV. Okay, makes sense.
So we've added to our test menu to comment on that. Whereas we look at bacteria through the test entitled MyPeriopath, when we add a human genetic marker, which helps us to characterize or catalog the robustness of a patient's immune response, then we have a fuller picture. How much bacteria are involved in these infections and how robust is the patient's immune response to keep those infections in check. That's the Alert 2 test. Now, not to be overlooked, there is an epidemic of oral cancer, and that's almost a topic unto itself for a podcast. We are particularly expert in the understanding of HPV, human papillomavirus, and its infection in the mouth, the pharynx, the larynx, as a cause of the majority of oral cancers. And so we have used our test to help dentists partake in this discovery of patients who have early oral cancers. To be clear, not different than the periodontal discussion, infection with HPV happens well in advance of there ever being an obvious lesion and hence a cancer in a patient's mouth. Our goal is to help dentists really be part of that preventative effect, finding infections, hence finding people at risk for these oral cancers well in advance of them seeing a horrible lesion. That said, we also get a lot of case material in consultation where a surgeon has removed an enlarged lymph node that shows squamous cell cancer, and we somewhat uniquely are capable of showing that HPV is present or absent in that cancer. That's really important for the selection of therapy and for prognostication for that patient. So oral HPV is part of the other things that happen in the mouth besides periodontal disease. And last but not least, you'll see a test very similar to MyPeriopath, except it's focused on different bacteria, the bacteria that cause dental tooth decay or caries. Different bacteria. So we're measuring that quantitatively to help dentists appreciate who are the people at risk, who harbor the bacteria that causes that acid deposit on the surface of the tooth. That, along with diet, looking at the patient directly and using different types of stains and dyes, you can anticipate dental caries. Well, our test actually finds the people who are carrying the bugs in their mouth well in advance of them having these cariogenic lesions.
Yeah, I've got a quick question here. I guess so far, knock wood or knock enamel, I'm one of those people that, pretty much has like no cavities, and my wife hates me for it and all that, but she has, had plenty of cavities throughout her life and all kinds of difficulties. And I asked my dentist somewhat recently, and he said, oh, well, it seems to be a dichotomy. People that don't get any cavities have certain problems, and then people that get a lot of cavities have other problems. Do you have any knowledge of what that dichotomy is, and on a bacterial level, why that would happen, and you'd have two different types of people with dental health?
I'm not entirely sure I know how to respond to that, except that I've heard some dogma, and I don't know if it's substantiated, that people who have a lot of cavities tend not to have a lot of periodontal disease. I can't say I can answer that honestly. All I know is that periodontal disease is a combination of the bacteria plus the host response, the inflammatory host response. Some people are resistant and some people are sensitive to it. The same tension, if you will, applies to patients who have dental caries. There clearly is a genetic predisposition. I don't believe we've honestly identified what genes are involved in the susceptibility to caries, but the combination of having the bacteria plus that genetic proclivity is the patient who has a lot of cavities or a lot of periodontal disease, as it were.
Okay. Yeah, well, it seems like you know something about it, but okay. Well, are you guys doing any of your own clinical trials, or is it plenty, with all the inbound test requests for people that are doing trials and various studies to elucidate more of what's going on in the mouth?
The answer to the question is yes. So we continue to do our own self-supported clinical studies towards the goal of finding more intelligent ways to analyze this data and to package it in ways that can help dentists to be more preventative or to know which course a patient is on. And I described that somewhat earlier to you, this ideogram, by taking serial measures with our test. What does that profile look like for a patient, but what does it look like for a typical patient of disease severity, mild, moderate, or severe? But the next step sort of beyond that will be to find ways in which we can make the use of this test simpler. That involves applications of doing the test at home, which is not to say that we're going direct to consumer, because that is not allowable at this point in time, but rather to see if the test taken at home is of the same quality as if it was collected in the dental office. This can operationalize the use of that test for a lot more patients. It's simple, the information is there when you come to visit, and we can act upon it immediately rather than waiting 2 or 3 days for the result to come back. So those are the kinds of directions we're going. But I guess to the first part of this interview, which was we're also looking for other types of tests packaged in simpler or more complex forms that provide more information. Case in point, what do I do, for example, with a patient who has an unexpected bacterial profile when the clinician tells me that they have severe inflammation, bone loss, and there's evidence of the risk of tooth loss? That does happen. Is our test wrong? No. It could be that there are other bacteria, which we happen not to be measuring, that might be involved. But we also have found that it's not always bacteria. For example, we have a goodly number of cases now demonstrated that flare-ups of herpes simplex virus. We know it in the form of aphthous ulcers or canker sores, sometimes occur deep in the periodontal pocket. When that inflammation arises, the tooth is made vulnerable and the periodontal disease gets worse. So we've been advising clinicians of late not to just think that the test is there to simply confirm that which they see with their eyes, but sometimes they need to go to a second tier or a third tier of testing to really find the true culprit. And this is the right way to use the clinical laboratory, an algorithmic approach that common things happen commonly, and we have a library of backup tests to help eventually find the culprit. And we spend a lot of time working with our professional colleagues in explaining this use of the clinical laboratory. It's the most economical and judicious way to do it. And as a background, I'm a pathologist, so it's about how the laboratory can contribute every day to the care of patients. Not always to give you the yes answer, but sometimes to create the scenario that I need to look further, look for additional information through other tests.
Okay, no, that's fair. Well, very good. Well, Ronald, we're coming to the end of time. What's the best way for people to find out more? And if they go to their doctor, they go to their dentist, periodontist, whatever, to say, hey, do you know about these tests by oral DNA labs, and can you order them for me because of X, Y, or Z? Like, how can people take action if they're concerned? Where do they go from here?
Simplest thing to do is to go to our website, www.oraldna.com, oraldna.com. We have an extraordinary customer service department who is very personable and will speak to any level of questioning. But the simplest first step for a patient or an interested person is that we can use our doctor finder and find a provider in their locale, and that's where the conversation can continue. For dentists or healthcare professionals interested, same process, reach out to us through our customer service. We can create an account online in just a matter of a few minutes. We make test kits available to your office. They come at no charge to the office until the test is actually ordered for a patient. And then it's all done electronically through a simple registration through a web-based delivery of the report. But I think equally important to our business, which is running laboratory tests, is the fact that we are here to provide a lot of consultation to a profession that is relatively new to the use of the clinical laboratory. It's important to allow other professional specialties in so we can put the pieces of the puzzle together, and just start at the website. That's all I can offer up.
Oh, that's great. Okay. Well, very good. Well, Ron, thank you for coming. It's been a very interesting call. I appreciate it.
My pleasure. It's a pleasure speaking with you, and thank you for your insightful questions.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.